Let's take a look at the efficacy of DNA replication. The rate of DNA replication varies significantly between organisms. For instance, in E. coli, the rate is approximately 500 nucleotides per second, whereas in mammalian cells, it's around 50 nucleotides per second. Such high rates of replication necessitate a continuous interaction between the DNA polymerase and its template. To achieve this sustained attachment, specialized proteins facilitate the continuous binding of DNA polymerase to its template. Consequently, DNA replication must be processive. To illustrate the concept of processivity, let's consider a simple example. Imagine you want to create 100 strings of pearls, which each contain 100 pearls. You have two options to complete this task. In the first option, you add one pearl to each string sequentially. So in the first step, you add one pearl to string 1, in the second step, you add one pearl to string 2, and so on until you reach string 100. After completing this round, you go back and repeat the process, adding a second pearl to each string, continuing until all strings are complete. The second option is to complete each string before moving on to the next. In the first step, you fully complete string 1 by adding all 100 pearls to it. Then in the second step, you move on to string 2 and add all 100 pearls to it, and you continue this until all 100 strings are finished. The first option represents low processivity, where you handle one pearl, or nucleotide, at a time across all strings, or DNA strands. The second option, however, illustrates high processivity, where you complete each string, or DNA strand, in full before moving on to the next. This analogy helps to illustrate how processivity relates to the efficiency and speed of DNA replication in biological systems. In the context of DNA replication, processivity refers to the number of deoxyribonucleotides that DNA polymerase adds to a growing DNA strand before dissociating from its template. High processivity is crucial for the fast and efficient replication of a genome. Achieving high processivity involves tethering the core DNA polymerase to its template. In prokaryotes, this function is fulfilled by the DNA sliding clamp, which is a dimer made of two identical subunits. In eukaryotes, the DNA sliding clamp, known as proliferating cell nuclear antigen, pCNA, is a homotrimer, which consists of three identical subunits. These sliding clamps encircle the DNA strands and ensure that the DNA polymerase remains attached to the template. This facilitates rapid and uninterrupted DNA synthesis. Let's look at the prokaryotic DNA polymerase III holoenzyme. The holoenzyme includes the core enzymes alpha, epsilon, and theta. The holoenzyme also includes two beta subunits, which form a sliding clamp that wraps around the DNA strand. This beta sliding clamp interacts with the core enzyme and tethers it to the DNA template. On its own, the core enzyme's binding to DNA would be unstable. The addition of four other subunits, including the gamma subunits, forms the DNA polymerase holoenzyme, enhancing stability and functionality. In the case of eukaryotic DNA polymerase delta, the holoenzyme includes polymerase delta 1, which is responsible for both the catalytic and exonuclease activities. It also contains Pol delta 2, P50, which interacts with the catalytic subunit. It also contains Pol delta 3, P66, which interacts with Pol delta 2 and is necessary for the recruitment of pCNA. The pCNA interacts with the holoenzyme and ensures efficient attachment to the DNA template, thus facilitating high processivity during DNA replication. When it comes to eukaryotic DNA polymerase epsilon, the holoenzyme is composed of several key subunits. The primary catalytic subunit, Pol epsilon 1, P261, is responsible for both the catalytic and exonuclease activities, as well as processivity. Pol epsilon 2, P59, is tightly bound to the C-terminal end of the catalytic subunit, which stabilizes it, and it interacts with pCNA, the sliding clamp that enhances processivity. Additionally, Pol epsilon 3, P12, and Pol epsilon 4, P17, interact with each other and the C-terminal domain of the catalytic subunit, which further contributes to the overall stability and function of the holoenzyme. The processivity of DNA polymerase epsilon depends on three events. The first, which is the only one shared with DNA polymerase delta, is the recruitment of pCNA. Next, the additional P domain of Pol epsilon 1 wraps around the DNA, which enhances processivity by ensuring a more secure attachment to the DNA strand. Finally, the subunits Pol epsilon 2, Pol epsilon 3, and Pol epsilon 4 form an extended domain connected to Pol epsilon 1. This domain forms a flexible tail that can interact with the DNA template and switch between open and closed conformations. In its closed conformation, this complex forms a groove that can accommodate up to 40 base pairs of DNA. This significantly reduces the risk of the DNA polymerase dissociating from the DNA template, which thereby increases the processivity of DNA polymerase epsilon. In conclusion, processivity in DNA polymerase is ensured by mechanisms that promote the sustained attachment of the DNA polymerase to its template. This is achieved by the formation of a sliding clamp associated with prokaryotic DNA polymerase III and eukaryotic DNA polymerases delta and epsilon. Additionally, DNA polymerase epsilon has unique features that further enhance its processivity, including a P domain that wraps around the DNA and an extended domain formed by subunits Pol epsilon 2, Pol epsilon 3, and Pol epsilon 4. These features, along with the recruitment of pCNA, significantly reduce the risk of the polymerase dissociating from the DNA template, which ensures efficient and accurate DNA replication.